So there are the three domains, eukarya, archaea, and bacteria. So the ancient life gave rise to the split between bacteria and eukarya sometime a little bit greater than 3 billion years ago. So that was when the ancestor of all life existed. Um, and then in the lineage that has eukaryotes and archaea on it, sometime greater than 2 billion years ago, the split between eukarya and archaea took place. So on that lineage to eukaryotes are the innovations in cellular structure that we're going to talk about. They all happened on this lineage sometime greater than 1 billion years ago. So here are the major innovations. First, there was the loss of the cell wall, loss of the prokaryote cell wall, which allowed the infolding of plasma membrane, increasing the surface area of the cell without increasing volume. There are prokaryotes with complex folding of the plasma membrane, but it's believed that the loss of that prokaryotic cell wall really enabled these eukaryotic lineages to really develop fundamentally new structures that became structures in all eukaryotic cells, such as the Golgi apparatus and the endoplasmic reticulum, that really enabled fundamentally different structures in these cells, freeing up the plasma membrane from the cell wall. Another membrane-bound structure is the digestive vesicles that are present in eukaryotes. So those were gained as part of this greater complexity of membrane structures. There's a more complex flagella structure, fundamentally different, fundamentally larger than the prokaryote flagella. It's actually a part of the cytoskeleton and surrounded by the membrane. We'll talk about that. Nuclei, membrane-bounded structure containing the genome. That's what eukaryotes are named after. Carry refers to the genetic material of at the cell biology level, the staining structures that are the chromosomes. They turned out to be the genetic material as cell biology advanced in knowledge. U meaning good. So eukaryotes are those things that have good nuclei. You can see by staining the cells. The organelles, the main ones we're referring to when we say organelles are mitochondria and chloroplasts. There are some other structures we won't talk about that also get called organelles, but those are the main two. So almost all eukaryotes have mitochondria. There's a few that have secondarily lost mitochondria. And chloroplasts have evolved multiple times. They're present in a number of lineages in eukaryotes. So there are primary, secondary, and even more complex events, and we'll talk about those events. So this is a schematic of what happens with the loss of the cell wall. You have a cell that's spherical, but within that sphere you can have a great increase in the amount of cell membrane since it's no longer associated with the cell wall. So you can have infolding, which greatly increases the surface area without increasing the volume. And the surface area is where a lot of the action of the cell goes on in terms of its communication with the outside world, in terms of its functioning of what types of things that cell does. So it enables a much greater area for those functions, for those exchanges with the outside world to take place. And it also enables the evolution of membrane-bound structures within the cell, such as the endoplasmic reticulum and the Golgi apparatus and other structures that you will study in a cell biology course. So it's a little bit about cell walls. They're present in prokaryotes, so virtually all prokaryotes have a cell wall, and there's structural diversity within prokaryotes, especially in the archaea. We won't talk about that. You would hear about that in a microbiology course. The ancestor of all eukaryotes, as we've been saying, lost the prokaryote cell wall. And then specific lineages of eukaryotes, namely plants and fungi, independently evolved a new cell wall. In each case, they're fundamentally different, both from each other and from the prokaryote cell wall. So they're still called the cell wall, but they're really different. So that's important to keep in mind. So the major, you could say, other than the nucleus, the major defining feature of the eukaryotic cell, or one of the big major ones, is the mitochondria. The singular of mitochondria is mitochondrion. And it's the result of an invasion of a prokaryotic cell from the proteobacteria lineage at the base of the eukaryotic clade. So the progenitor of all eukaryotes became host to this proteobacteria, which lived inside of its cytoplasm and evolved into mitochondria, a structure that's used for making ATP for energy. It's also involved in many other cellular processes, such as growth, signaling, and programmed cell death. Here's an electron micrograph of a couple of mitochondria from the mammalian lung tissue. You can see the mitochondrial matrix there and the membrane structures. These are in the cytoplasm, and they're maternally inherited because the cytoplasm of a zygote comes from, at least in anisogamous sexually reproducing animals, comes from the mother. So, and they will talk about this theory that led to the conclusion that mitochondria are evolved from this proteobacteria. That the thing in recent times, in modern times, in the last few decades, that has really confirmed this hypothesis, which was originally from structural data, is that there's DNA in mitochondria. They have a genome, and when it's sequenced, the sequence turns out to be similar to proteobacteria, not to eukaryote nuclear DNA. There's been a lot of genes lost from that compared to what the proteobacteria has. Some genes have been moved to the nucleus through evolution, but the DNA that remains in mitochondria looks like proteobacteria genes. So this phylogenetic tree depicts the invasion of mitochondria into the early eukaryotic lineage, and we don't really know. It's got to be sometime very early in eukaryotes, because that lineage gave rise to all the eukaryotes we have, but time isn't really depicted on this phylogeny, so we don't know when that was. We know that it was before chloroplasts came in, because all eukaryotes have mitochondria, but only some lineages of eukaryotes have chloroplasts. So those lineages must have diversified before chloroplasts came in. There was a single origin of the first chloroplast called the primary event for chloroplasts, the primary endosymbiotic event, and that came from a cyanobacteria. And that primary type of chloroplast is present in land plants. It's the chloroplast we're most familiar with. We'll talk about the later events that gave rise to different types of, more complex types of chromosomes in the next slide. So mitochondria evolved once. Chloroplasts, the situation is more complicated, but there was a single primary event resulting from endosymbiosis, a cell living inside another cell. In the eukaryotic lineage, this was the origin of all other chloroplasts, all the chloroplasts. Many lineages have chloroplasts from eukaryotic symbionts. So eukaryotic lineages went and invaded other eukaryotic cells, and they became the chloroplasts of those cells. So when that happened the second time, it's called a secondary symbiosis, and there are tertiary and quaternary events too. So this slide depicts that. Here's on the left is the primary event. There was a cyanobacteria invading a eukaryotic cell, and it came to live within the cytoplasm of that eukaryotic cell. So you have the host cell has a nucleus, but it also has this membrane-bound structure, the chloroplast, which actually has a double membrane. One membrane from the plasma membrane of the host cell, and the other membrane which was around the cyanobacteria originally. So it has a double membrane. And then in secondary endosymbiosis, a lineage of this eukaryote this now photosynthetic eukaryote with a primary chloroplast inside, invades another eukaryotic lineage, in an event, and that before this event, that eukaryotic lineage did not have chloroplast, but now it does. But now it's a more complex kind of chloroplast, because it's not directly from a prokaryotic cell; it's directly from a eukaryotic cell with a chloroplast in it. So that's a secondary event, and you've got another membrane structure, another membrane added around it. So there's an addition of a membrane. And this can happen again, and you'd have multiple. You'd have a tertiary event or a quaternary event. And there's evidence for tertiary and quaternary events where this eukaryotic cell on the lower right goes into another eukaryotic lineage and becomes a type of chloroplast. So these get very complicated, and if you take a microbiology course, you would study this more. That's as much as we're going to say about it now. Just to know that chloroplasts, and more generally they're called plastids, have evolved multiple times, and they generally do photosynthesis. Their photosynthetic pigments are similar to the original cyanobacteria, which is the clue that really there's a single origin of all chloroplasts, this original primary event that's depicted on the left. We can tell when you look at the cells of a photosynthetic lineage of eukaryotes, you can tell what type of event it was from the number of membranes and some other structures that are there. So this cartoon depicts the primary event, which went into a single eukaryotic lineage. That lineage diversified into a lineage that became the green algae in plants, and we'll hear a lot more about that lineage later in the course. And another lineage became the glaucocystophytes, another group, and another lineage became the red algae. The red algae lineage then invaded some other eukaryotic lineages to make a bunch of secondary endosymbionts shown down here. We're going to talk briefly about some of those protist groups down here in the bottom in purple. But also this primary chloroplast is a chloroplast in green algae in plants, but lineages of that group went into some other eukaryotes and then became the chloroplasts that are present in euglenids and some other groups such as the chloroactinophytes, which we won't talk about. So there's a lot of groups here. We'll only talk about a few of them. So how do we get all these plastids? So first and most important, you have this primary event that came from a cyanobacteria. The original host, the eukaryotic cell, had mitochondria. It was a eukaryote, but it didn't have chloroplasts. It wasn't photosynthesizing. It became photosynthesizing. So the ancestor of all the other chloroplasts in the tree of life is this original primary event that took place once in a eukaryotic lineage. So, and all the other chloroplasts were lineages of this that descended from this event that went into another eukaryotic cell to give rise to the secondary, and then after that tertiary events, etc., getting complicated. So why did this happen? Think about life in the water, which was all life until invasions of land that we'll talk about later in the course, involving plants, involving invertebrates, and involving vertebrates, much later in the history of life. Life was in water. So cells and organisms in water are eating other cells and organisms. So instead of digesting a cell that you just engulfed, maybe it lives for a while. So maybe it survives for a while and actually provides some benefits. So if you have a photosynthetic cell inside your cells and it's doing photosynthesis, it actually might be able to provides some carbon to the host cell. So eventually the hosts become dependent on this. This increases the fitness of the host. The host can survive better and survive for a long time and then eventually become dependent on this relationship. This is called endosymbiosis. We talked about symbiosis, two organisms living in close contact with each other, and there's usually some plus-minus relationship to this. And if it's a long-term one, it's often a mutualism where both the host and the invading cell have a benefit. Endo because it's actually inside the cells of the host. So the benefit to the host in both mitochondria and chloroplasts is energy, and in terms of chloroplasts, it's fixed carbon due to photosynthesis. The benefit to the little invading cell is shelter and food from and nutrients and stuff from the host cell. So it's a mutualism. So the theory is over 100 years old now, and it's called endosymbiotic theory. The theory that mitochondria evolved from ancient bacteria and chloroplasts evolved from ancient photosynthetic bacteria or photosynthetic eukaryotes. This is the first person to put this forward was Konstantin Markovsky in 1905, and it was originally thought to be a pretty crazy idea, but he was looking at cell preparations and the structures and suggesting an explanation for why these structures that had chlorophyll in them had these complex membrane structures around them. So during the middle part of the 20th century, Lynn Margulis revived and more fully investigated this with much better tools and better data in the 1960s and 1970s, and she really put this idea on the map and put all the evidence together. So what was once thought to be a strange idea with only structural evidence, in the 60s, 70s, and 80s got DNA evidence which sealed it, which made it textbook knowledge that this is where these structures came from. Margulis became famous for this. She actually just died a year or two ago. So another fundamental eukaryotic structure is the cytoskeleton. It's made of monomers that can rapidly polymerize and depolymerize. The main ones are actin, tubulin, and intermediate filaments. So these three stainings over here on the upper right are the three different types of polymers that you can stain for inside the cell that make up the cytoskeleton. So this is important for a lot of things that the cell does, for motility, for moving around. So if you're familiar with amoebas and putting out these little arms, those are called pseudopodia. That is all driven by movements of the cytoskeleton, the rapid polymerization and depolymerization of these polymers, of these actin, tubulin, and intermediate filaments. Also the cilia on the outside of cells, and the flagella, which we'll talk about a few slides from now, are functions of the cytoskeletal structure dynamically moving, polymerizing and depolymerizing. The chromosome movement during mitosis and meiosis is also controlled by the cytoskeletal movements. So on the right here we have, I believe it's meiosis, all those movements of chromosomes attaching to which attach to the spindle. The spindle is part of the cytoskeleton, and driving the whole process of the cytoskeletal movements. So some cytoskeletal proteins have recently been found in some bacteria, and a few years ago, Jared Postolia, a Bio 201 student, sent me this paper about the bacterial cytoskeleton. So cytoskeletal components have been discovered in prokaryotes. So some prokaryotes have their own form of cytoskeleton. So they have homologs, molecules that are related to actin, tubulin, and intermediate filament due to inheritance from a common ancestor. That's the definition of homolog. They also have a unique group of proteins in these structures called MIN-D and SOJ. Those are not present in eukaryotes. So there's also some evidence for these types of structures in archaea, but they're not as well studied as in bacteria. So this figure on the right just shows you that not only do you have some of these cytoskeletal proteins in eukaryotes on the left column, but also in prokaryotes and archaea. So how do bacteria use the skeleton? So the cell wall in bacteria is known to be the primary determinant of cell shape. But cytoskeletal elements might help regulate the formation of cell wall structures into particular shapes. And also during cell division, they might play a role as well, because the cell wall does have to go through some dynamic changes as cell division takes place. So the bacterial cytoskeleton might play roles in that. Also in cell polarity, making one end of the cell structurally different from the other end of the cell. And there's a lot of diversity in prokaryotic cells, indicating that they do have polarity. And also partitioning of where the chromosome, the DNA, is in the cell during binary fission, cell division of prokaryotes. So we need to rethink the role of the cytoskeleton in eukaryotic evolution. So the common ancestor of all three domains had actin and tubulin. That's the evidence. And it would probably be more informative to find out more about archaea, but we can say that about a lot of things. Archaea are still very mysterious. So the eukaryotic cytoskeleton really generates forces in multicellular tissue, so in muscle, and interacts with what's called molecular motors, which are these things that interact with the cytoskeleton in the form, and some of the types that are called kinesin, dynamin, myosin. Now these types of things have not been found in prokaryotes. So this is what multicellular eukaryotes do with many, many cells coordinating their cytoskeletal movements. The loss of the cell wall in the lineage that gave rise to eukaryotes provided evolutionary opportunities for more diverse functions of these cytoskeletal components. So that's a possibility. So all of this is kind of buried in the past, but from molecular data and cell biology data, we can get clues to what the process of evolution involved in getting to how the eukaryotes use what we formally call the cytoskeleton. So here's a picture of the bacterial flagellum. It's a single fibril that projects out of the membrane of the cell wall. It's composed of flagellin, a protein. It's got a complex structure with all these components here. Don't worry about memorizing those. The hook and basal body drive the motion of this thing. The eukaryotic flagellum is much bigger. It evolved independently, sometime during that eukaryotic, the ancestor to all eukaryotes. It's enclosed by the plasma membrane, which is depicted in this cartoon here, and it's got a lot of things inside it. It's got 9 pairs of microtubules surrounding 2 pairs of microtubules in the center. You can see in these cross sections. So there's a more distal cross section above and a more basal cross section there. So it's a very large structure, a very complex structure. It's not this single fibril. It's really an extension of the cytoskeleton surrounded by the membrane. So very different. So there's huge diversity in single-celled eukaryotes, and a general term we use for single-celled eukaryotes is protists. So protists are eukaryotes that are not plants, not animals, or fungi. So for an introduction to protists, there's a video there that you can see. We won't show it in this video. There's vast diversity in reproduction, in metabolism. There's phototrophs, there's heterotrophs. In the video, they call them organotrophs. And there's vast diversity in morphology, even among just single-celled eukaryotes, the protists. So, but many protist groups can also be multicellular. The multicellular groups species are generally colonial. They live as colonies, and they don't have what we call the tissue level of organization. But there's still a lot of complexity there. So let's go through some terms here. So protists, again, are eukaryotes that are neither land plants, animals, nor fungi, and this term is still used. It's an old term, but it's still used. An old term that's not used very much is protozoans. It's got this root zoan in there, which suggests animals. So this used to be the term for microbial eukaryotes classified as single-celled animals, but we now define animals as multicellular. Another word for animals, metazoans. So protozoans is not a useful term. It's not very used much. Protist is a more useful term. Now, we talked about in taxonomy and phylogenetic systematics, wanting to name groups, to recognize groups that are descended from a single common ancestor, and not recognizing non-monophyletic groups that don't include all of the descendants of a common ancestor. So both of these classifications, protists and protozoans, do not include all of the descendants of the common ancestor. The common ancestor would be the ancestor of all eukaryotes. Because we're talking about mostly single-celled groups here, they're also mixed in that diversification with multicellular groups. And so just to use the term protists, we're talking about a mixture of lineages that are not, and we're excluding lineages that are related that are multicellular. So we'll learn more about phylogenetics and monophyletic groups later in the course, but from that perspective, this term protist is a little bit problematic, but it's still useful. So just a brief review. So we know what diploid and haploid cells are, and we're familiar with mitosis, in which a diploid parental cell duplicates its DNA. So the chromosomes duplicate, and then in mitosis, there's one cell division leading to identical daughter cells. So it's important to note that this happens, we're familiar with it in diploid cells because that's what mitosis is for us. But also in organisms with a haploid part of their life cycle, haploid cells can also duplicate via meiosis. So the important thing about mitosis, via mitosis. The important thing about mitosis is that it creates identical cells. You know, we'll set aside the possibility of somatic mutation, which always takes place, but setting that aside, mitosis generates identical cells, and it doesn't change the ploidy of the cells. So a diploid cell can go through mitosis and yield identical diploid cells. A haploid cell can go through mitosis and yield identical haploid cells. In meiosis, you have diploid parental cell duplicating its DNA and then going through 2 divisions to lead to 4 non-identical haploid progeny cells. So the non-identical thing because of recombination and independent assortment of multiple chromosomes, but haploid because you have 2 cell divisions. You have a reduction in ploidy. So importantly for these life cycles that we'll talk about, meiosis involves a reduction in ploidy from diploid to haploid in the simplest case. So there's a lot of diversity in reproduction in protists, and we don't have any time to talk about it. You could do an entire course on that by itself. But just to show you how weird things can get, this is Paramecium, a well-known ciliate, single-celled eukaryote protist. And this is conjugation. It's sexual, but it's not reproductive. So what you have is you have 2 cells getting together through signaling. They're in the water. The genome of these cells is present in 2 forms in each cell. The macronucleus, which sort of runs the cell, and the micronucleus, which is involved in reproduction, or involved in genetic exchange, as we talked about here in conjugation. So there's multiple micronuclei when the cell is just out there in the water. In advance of this event, all but one of those micronuclei disintegrate, leaving just one. The remaining one undergoes meiosis. Okay, so meiosis within the cell, meiosis without cell division, making nuclei. So 4 haploid nuclei are the result of this meiosis in each one of these cells. 3 of those 4 disintegrate, leaving 1. And that one undergoes mitosis to duplicate, and then these 2 cells that have joined will exchange one of those. So this is the sexual exchange, but there's no reproduction of new cells here. So then those 2 micronuclei in each cell, they're genetically different. Those fuse, and then they make essentially a new recombinant micronucleus. Those divide mitotically, and eventually they give rise to a macronucleus that's again a copy of this recombinant micronucleus. The 2 cells that divide or separate from each other and go their own way, and then through asexual type reproduction they can make more and more cells. So the reproduction takes place somewhere else. This is the genetic exchange. Now this is very bizarre, very complicated. I don't expect you to memorize what's on this slide, but I went through it just to illustrate how complicated it can get. A lot of cell behavior adaptations here to do this process of sexual reproduction. And remember, we talked about the advantages of sexual reproduction. So this is just an illustration of how some species really go to great complicated lengths to take place in a sexual exchange here with other cells. So that's just one of many different types of sexual reproductive processes that take place among protists. So here's a general depiction of life cycles. So we have a change, a constant cycling between haploid and diploid. So you have, this is called alternation of generations between haploids and diploid. So there's a gametophyte that's haploid, and there's a sporophyte that's diploid. To go from the sporophyte to the gametophyte, you go through meiosis. And then you have a haploid part of the life cycle where you have mitosis, reproduction of haploid cells. And then to get back to the diploid phase, two haploid cells get together in a fertilization event. And then they reproduce during mitosis, and then we're back around. We have a multicellular diploid organism, and that some cells of that organism undergo meiosis to make new haploid cells. So land plants do this. Many multicellular photosynthetic eukaryotes do this, and we actually call this a sporophyte and a gametophyte. So they have established, and in some cases long-lived, both phases of sporophyte and gametophytes. This is unfamiliar to us because we only have a very brief haploid stage, our gametes. But many, many other organisms have a long-lived, established multicellular haploid phase, the gametophyte. So let's look at some of the, a couple of lineages of algae that have some variations on this. This is the sea lettuce, Alva lactuca. It's a chlorophyte. It's a type of algae. So you have the haploid and the diploid phases. You have a multicellular organism that looks pretty much the same unless you put it under a microscope in both phases. And if you're just looking at it with your eye, you can't tell whether you're looking at the haploid phase or the diploid phase. In the haploid phase, it looks like a sea lettuce. There's mitosis going on that's producing gametes. I mean, this whole thing in the top is haploid. Those things will fuse. Two gametes will fuse to make a diploid zygote. Now you're in the bottom part of the life cycle. This will undergo mitosis to make this multicellular diploid. And in some tissues of that, at the tips of the leaves here, you have meiosis going on to make new haploid spores, and you start the cycle all over again. So when the male and female gametes are not different, again, unfamiliar to us because it's not sperm and egg, but it's genetically genetically equivalent to sperm and eggs, but they're not different. This is called isogamy. Okay, so not all. And then when you have two halves of the life cycle, one haploid, one diploid, they're approximately half, and they look the same in the two different halves. That's called isomorphic. So Lactuca here is isogamous. The male and female gametes are not different, but its life cycle is also isomorphic. It looks roughly the same between the haploid and diploid phases, and the haploid and diploid phases are both there for a while, although it might not exactly be 50% each for the time of the life cycle. Another term here related to the gametes, when you have sperm and egg, when you have really different gametes, that's called anisogamies, and that's the situation that we're familiar with. Now here is a life cycle of Ulothrix, another chlorophyte, where the haploid phase dominates. So this is called haplontic life cycle. There's only a very brief diploid phase where there is a zygote, which immediately after fertilization undergoes meiosis. So there's no multicellular diploid in this life cycle. Almost all the time it's in this haploid life cycle where it has a multicellular form. It can actually undergo asexual reproduction in that multicellular form and make new strands of cells here, but then some of the cells in each strand are going to become gametes, and there will be fertilization to form a new zygote. So that's a haplontic life cycle. Most of the life cycle is in this haploid form, and there is no diploid multicellular form. So what are we? We're the opposite of that. We're diplontics. We have a long-lived dominant diploid form, which you could call the sporophyte, and the gametes are this very brief phase that basically terminates with fertilization. You have a zygote, and then you're back to the diploid phase. There's no multicellular haploid phase. There's just the brief gamete phase. So that's the diplontic life cycle.